Before I start my talk, I will show you the view of our university. I'm in the Kimcheon University. This is Taiwan, and I'm from South Korea. And Kimcheon University is up here, under the center part. This city is the center of Taiwan, yeah? Is it right? Yeah. And the Kimcheon is almost the center area of the South Korea. And in Korea, and the Oriental sayings that if there is a good cast, there is a big rain, right? And the typhoon is coming, so I really deeply thank you for the Taiwan to be welcome. Anyway, the topic of my talk is FS2. FS means follicle stimulator, and its main ingredient is NXCL or 4950. 4950 is number of our trial to found this compound, and its second stage new formula is NXHRC. It developed last year, so we put the last year number. As I told you, as mentioned, I am from South Korea, and historically, Taiwan know about the South Korea as a TV drama. 여장교, 매력성 그대, and the Gangnam Style. But not only the cultural interaction, historically we have a deep interaction between political, CKS. We also learned our international history. CKS as a founder of Taiwan and as a good leader of Taiwan political history. And he's our first president, Dr. Lee. And he visited Taiwan in 1953. It's the end of the Korean War. And they have a good relationship. And we also learn this story in our history class. And also, CKS and Park Chung-hee, he's the father of Park Geun-hye. She's now our president. And he also made a good relationship between the CKS. And he visited here in 1966. And now the Prime Minister, President, how come? I don't know. Anyway, the Maiju, is it right? Maiju? Yeah. We also learned about him. This is former President Lee Myung-bak, and we compare two guys as a very similar. We found similarity. So for newspapers, and we talk about Maiju as Mr. Lee Myung-bak. Anyway, so I think not only the political history, now many leaders in economy, we have a good relation with Taiwan. And today, I've come here to make a good scientific relation between you and us. So that's why I show you these pictures. Okay, today I'm going to tell you about hair loss treatment by our technology. Do you know who he is? Many soccer fan, Robben. Yeah, he's a famous player from Holland, Netherlands, and he's a representative athlete of Netherlands player. And he now is a Bayern München in German Bundesliga. But he's only 20 years old, 29 years old. But he looks like over 50, right? Because it's because of his hair loss problem. And when he was interviewed with the press newspaper, he said he tried everything he can do. Because he has a lot of money, so he can buy anything and he can try everything, but he could not solve the problem. If he meet TSTSM, I don't know. And this society he can't solve easily, but anyway, now he's still bored here. As you know, the hair growth stage has 4 steps: anagen, catagen, telogen, or anagen. It goes a cycle, and it takes from anagen takes 3 or 5 years, and then almost 2 weeks, 4 months, and then you go back to the anagen. And hair loss starts various reasons. As you know, hormonal reasons, psychological stress, or some drugs, toxic drugs, or post-acute illness. Every reasons are complexly and do harm to and induce the hair loss. And you already know the DHT, dihydrotestosterone, is a major cause of the hair loss. And many companies and many researchers are focusing on to inhibit 5-alpha reductase for the target of solving the hair loss problem. DHT is the main cause of inflammatory reaction around the hair follicles. So if we block or if we remove the production of DHT, it could be a very good method to reduce hair loss. And our team focused on the hair follicle blood circulation. So we decided to develop a new technology to increase directly the blood vessel on the follicle, and then the activation could remove DHT, and we can give more oxygen and nutrient to the hair follicles. So this is our target. Blood circulation has many effects. You know, this is Raynaud's disease and rheumatism, and hair loss is a representative disease related to blood circulation problem. We try to recover the blood vessel contraction. You know, many psychological stress hormone can induce the contraction of blood vessel. Blood vessel contraction starts by the stress. The stress pathway starts with production CRF in the hypothalamus, and then it activates the pituitary gland under the hypothalamus, and pituitary gland makes ACTH hormone, and it arrives to the adrenal gland, and then ACTH activates ACTH receptor, and the receptor produces the cyclic AMP by activating adenocyclase. And the second messenger, cyclase, now activate protein kinase A, and then the cholesterol turn into the cortisol in the mitochondria. In the mitochondria, the cholesterol converted into the progranulin in the cytochrome P45, and then, do I have to finish? And then this cortisol will be released from the cell to the blood vessel. And many Raynaud disease and blood contracted patients try to overcome that kind of physiological problem. They wear the hot gloves or hot pack to use, but it's very acute effect. There is no chronic effect. So sometimes some cosmetic company uses capsaicin, you know, that it's in the hot pepper, or they use vinyl butyrylation to stimulate the sensory neuron in the skin cell. But it's very toxic, and it induces many side effects, and it feels very uncomfortable when you apply on your skin. So we want to make something to substitute that kind of non-scientific compounds. So we focused on the blood contraction mechanism. As you already know, the myosin and the actin filament must be bind to contract the blood vessel, and then the muscle must be vasodilated when they are binding are ended. Especially the myosin has a light chain. The light chain will be phosphorylated by the phosphate kinases, and then if the phosphate is binded, the myosin could be bind to the actin. If the phosphatase release the phosphate, it changes structure, and then the myosin will be released from the actin. The blood vessel turn into the vasodilated, relaxed. So this myosin is the final target of our research. This is before the activation. So they use ATP, then the myosin light chain kinase and calcium will activate this protein to binding to phosphate in here, and then the phosphatase will release this phosphate. So the blood vessel will be relaxed. So upstream of the signal transduction, there is a good target molecule, as you already mentioned, as I already mentioned, the adenine cyclase. This enzyme converts ATP to the cyclic AMP, and it activates protein kinase A. And this catalytic subunit of protein kinase A will dephosphate myosin light chain kinase. And if there is no cyclic AMP, the blood vessel is going to contract it. But if there is a cyclic AMP, the blood vessel will be relaxed. And the second target is guanine cyclase. It is also producing the cyclic nucleotides, and cyclic GMP will be activate protein kinase G, and it will activate myosin protein phosphatase, and then smooth muscle will be relaxed. So FS2 active compounds are designed to activate guanine cyclase and adenine cyclase. These two compounds will activate, produce cyclic GMP and cyclic AMP, and then these will be relaxed by the dephosphorylation of the myosin light chain kinase, myosin light chain. Before we find the compounds, our team used the computer-aided drug design system. This design system helping us to reduce our finding the new compounds, and then it tells us in very short time we can find good candidate for the regulate the protein activation. Last year, Nobel Prize for the chemistry awarded to the 3 persons. They are specialists and professional researchers for the computation analysis. CADD is a very simple concept. You know, if we find a target protein and we input in the computer the candidate ligands, and then the computer will solve and calculate the best candidate for the drug to regulate this protein, and then it tells us many statistical and mathematical data for the finding good compounds. While the protein processing, they show the binding pattern of a candidate compound and target proteins. And this tells us 2-dimensional results, and it also shows us a 3-dimensional interaction between the protein and the amino acid residues. So we could find good chemical compounds from the 5 representative herbs. As you already know, the licorice, Curcuma japonica, and raspberry cinnamon is a good candidate herbs from. You can usually use this kind of for the food ingredient on the drug sources. So guanine cyclase and adenine cyclase is a 2 target, and these compounds also their interaction also be confirmed by using the X-ray crystal research with the Pohang Accelerated Laboratory. As you know, this X-ray beam can figure out how the compounds binds to the protein target. So we could get this kind of picture. These compounds directly binds and it activates this protein to make second messages. And the next step, we must make a cells to easily penetrate the skin, especially the scalp. And then we designed this kind of nanomycel formula, and then it helps to penetrate into the target cell. So it's very mimics the phospholipid in the cell membrane, and we used some steps to activate using the ultrasonic stimulation, and we made nanoscale micelles, and it's from 18 nanometer to the 100 nanometer, and our target compounds are in the nanovesicles right here, and we normalize the sizes and extrude, and then we tap some target chemical to reach to the target cells. And when we apply our compound, the outer skin, and under 1 minute, it penetrates easily the middle of the skin, and then after 1 or 5 minutes, it penetrates over the 90%. So successfully activates and enhances the blood circulation. And we also use high enriched concentration of this compound to our hands, and the color change. It's a high concentration, so it turns into too much red, as you see. And this is hand of a Raynaud disease patient, and we use infrared camera to check their circulation and their temperature. After the application, the circulation changed and enhanced. The skin temperature also increased. In our head, the ear is the coldest part of our body, and we applied our compounds on the ear. The ear color also changed immediately, and it's over the average temperature of our skin. It's very effective. And also, this is a microvessel in our finger, and this narrowed microvessel changed. They opened and they expanded their diameter, and the blood vessel goes well. So by this technique, our team got an award from Moscow and in the Korean Society. And the next concept is NXHRC 213 is targeted 5-alpha reductase inhibitor. So we also use the CADD technique to inhibit the 5-alpha reductase. So FS2 original is NXCL, and F2 beyond is NXCL plus NXHRC 2013. So it's also from Earth, and we try to check the red mouse and the hair growth well when we apply the NHSRC. So these are chemical compounds, and we found the 2 representative ingredients, and it also we synthesized in the nanomycels, and we found their structure. You can see these are before using FS2 and after use FS2, and there are many clinical data before and after. And these are 80 times magnified zoom of the scalp. As you can see, the diameter and the color of hair is more dark than changed, and the number of hair are grown. And a 250 times magnified picture of the hair scalp. It's very effective. So we dare to say the FS2 ingredient is very kind of evolution compound for the hair loss, to solve hair loss problem. And all the study were collaborated with this kind of world-leading scientists, and we also are assisted by the many companies. So thanks to them, and you can find if you type my name, you can see the reference papers related to these techniques. And this is my laboratory students, and So